Today we will be talking about point mutations, which is the substitution of one base into another base. First, let's talk about the two types of base substitutions, which are transition and transversion. Purines, which consist of adenine and guanine, can be distinguished by their two rings. A way to remember them is by pure as gold. Pyrimidines are thymine and cytosine, which only have one ring. The 4-point mutations are missense, which is when one amino acid changes into another amino acid. Read-through, which is when a stop codon becomes an amino acid, making the sequence longer. Silent mutation occurs when the base substitution still encodes for the same amino acid. Nonsense is when a codon changes into a stop codon, shortening the sequence. Here we are given a DNA sequence. In order to code it, we need to transfer it into an mRNA, which is done by changing all the T's into U's. For this video, we will be focusing on mRNA strands. So here's the final sequence we will be using to demonstrate point mutations. Before we get started, we need to correspond each codon with an amino acid. A codon contains 3 letters. In this picture, we have a dandelion that is long and has read-through mutation. The dandelion without any hair has missense mutation and was unable to function properly. The third dandelion has nonsense mutation, which has a codon change to a stop codon early on, so it doesn't have much hair. The last dandelion has silent mutation, which we can assume looks like a normal dandelion. For this sequence, there are 2 bases that can change for nonsense mutation to occur. But for this example, we will only be using the one to the left. UGG turns into UAG, which is a stop codon. Because the sequence has been shortened, the petals are scarce. The base went from G to A, which are both purines and therefore transition. Here in this read-through mutation, the stop codon UGA changes to UGC, which encodes for cytosine. This sequence is continuous until a stop codon has been reached. The petals here have overgrown and become longer. The base changed from A to C, a purine into a pyrimidine, so transversion took place. Next, we have silent mutation, which is when the mutation does not impact the amino acid sequence. AAC into AAT in DNA still encodes both for ASN. Here the petals stay the same. This is transition because it goes from C to T. Last, we have missense mutation, which is when an amino acid changes into another, which can impact protein function, or it can be a neutral missense mutation, which does not affect function. However, here the petals were unable to function and did not grow. AAC turns into AGC, which went from ASN into serine. A to G is transition. In this video, there is a donkey, 2 minis, and 2 disproportionate ponies. The pony here has normal sequence. The other pony looks exactly the same because the mutation did not change how she looks, so this is a silent mutation. The donkey has missense mutation, so the mutation did not function properly and changed it from a pony to a donkey. Then we have this big pony that has read-through mutation and has gotten longer and taller. Lastly, we have this tiny horse that has gone through nonsense mutation, which means a codon changed into a stop codon early on, making the legs of this horse short.